Well, hello. It's me in my natural habitat. So I had planned on making a better video at the school in front of the board like I normally do, but of course, COVID had other plans. So we are making the best out of what I have to work with, which is a laptop and my living room. So, um, Today, we are starting a new unit, Unit 5. Unit 4 was very short. It was only two sections long, 4.1 and 4.2. The other stuff that was supposed to be in Unit 4, we already talked about, so we don't need to talk about it again. But we are starting a new unit of Unit 5, and Unit 5 is all about the cell life cycle. So, what does that mean? What is the cell life cycle? So the cell life cycle is just what it sounds like. It's the life cycle of a cell. Just like you, cells have a life cycle. Um they are born, they grow older, they reproduce, they get even older, and maybe they'll reproduce a couple more times. Some more than others. Some reproduce a lot. Some hardly ever reproduce. But eventually they get old enough to where they die, and their offspring, the cells that they made, take their place. And that's just the circle of life. Just like with people, no different. So today we're learning all about that cycle, that life cycle of the cell. So I'm just going to be showing you pictures and talking about it. And the pictures that I show you and the words that I say are going to be enough to answer the questions that are in the assignment. Uh, so, we're going to go through this together. Um, let's start off with the pictures. So, first, I have to do a screen share with you. Okay. So, um, I'm going to pull up some pictures. And this first one here just kind of gives you an overall view of, well, that looks really bad, of the life cycle of the cell. Um, so here are all the stages of the life cycle of the cell. All right. So we start off with G1. G1 is the first stage of the cell's life cycle. The G stands for gap. So the reason why it's called G1 is because there's actually two what we call gaps. There's gap 1 and gap 2 down here. And the reason they're called gaps is because there's not really a whole lot going on during those phases. There's kind of gaps in activity. So gap 1 is the first one, and gap 2 later on we'll talk about. But this is where the cell is born, right here. And the cell in gap 1, the cell is basically just kind of growing and maturing into an adult cell. And then it gets to a point in its life, and this is different. This time period is different for different cells. Okay, some cells grow and mature much faster than other cells. Some cells will reproduce a lot. Some cells will reproduce very little. It just depends on the type of cell. Like your brain cells, they only reproduce once every several decades. So you may only get, 2 rounds of brain cells in your entire life. However, your cells that make up the inside of your intestine, they only last a few days. So they're constantly dying off and being replaced by other cells. So it's different for every cell, but they all go through the same stages, just at different speeds. Some much more than others, and some much faster, and some slower. Okay, but the steps are all the same. And in the first gap phase, or G1, the cell is just growing. That's why it says cell growth right there. Okay, not a whole lot's going on. It's just growing. And then when it gets to a certain age, um, it enters what's called the S phase. Now, S stands for synthesis. The word synthesis means to make. And in this case, what the cell is making is a new copy of DNA. That's why it says right here, DNA replication. So before a cell can split, the cell has to double the amount of DNA inside of it, so that when the cell splits in half, each half has the right amount of DNA. If the cell did not double the amount of DNA before it split, then it would split in half, and each half would only have half the amount of DNA it's supposed to have. And some cells in your body do that, but not these cells. This is mitosis. Okay, that's a different phase. That's a different process. Um, Whenever they split, and each daughter cell has half the amount of DNA that's supposed to have. That's another process that we're going to save for later. But for now, we're just talking about cells that want to make exact copies of themselves, so that when they get old and die, they have kids that will take their place when they're gone. Okay? All right. So, S phase, DNA is replicated. We double the amount of DNA inside the cell. Now, if you're going to double the amount of DNA inside the cell, you could assume that the cell is going to get a little bit bigger. And that's what happens during G2. G2 is gap 2. Remember, there's another gap in activity. There's really not a whole lot going on at this point. The cell is just preparing for mitosis. Okay, so this is just kind of an in-between stage between the time that DNA is replicated in the S phase, or the amount of DNA is doubled, and the time that the cell is actually going to split in half, where each half becomes a new cell. That happens in mitosis, where the cell splits in half. But before we do that, before we talk about that, I just want to make sure that you understand that G1, gap 1, S, synthesis, where DNA is doubled, and G2, gap 2, where the cell is preparing to divide and split in half. Those three together are collectively known as what we call interphase. Now, the word inter means between. So interphase means the phase between times where the cell is going through mitosis. So the cell is either going through mitosis, where it's actually splitting in half into two separate cells, identical cells. It's between the time that it's doing that and not doing that. When it's not going through mitosis, it's in what we call interphase. But when I say interphase, I could be talking about either G1, S, or G2, because all three together are known as interphase, okay? But when it's not in interphase, that means it's going through what we call mitosis. Now, mitosis is actually made up of several different stages. The easy way to remember it is PMATC. In other words, PMATC. Okay. So if you can remember PMAT, then you're okay. So just say it over and over again. PMAT, 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 PMAT. Okay. These are the stages of mitosis. And this is where the cell is actually going through the process of dividing. So P stands for prophase, M stands for metaphase, A for anaphase, T for telophase, and C is cytokinesis. All together, all of these are known as cell division or mitosis. Okay. Now, we're going to go through each of these stages individually in just a second: prophase, metaphase, anaphase, and telophase. And we're going to talk about what happens during each of those stages. Um, so let's do that now. Okay. So when this picture loads, these are the actual stages of mitosis. So at this point, the cell has gone through interphase, which are all three stages of G1, S, and G2 together. And the cell is now ready to actually split in half, and each half become a new cell. Um, the first stage of mitosis is what we call prophase. This is the P in PMAT. So in prophase, you need to know what happens during prophase. There's two very important things that happen. The first thing is you see how this line is dotted? This is the nuclear membrane. This is the wall of the nucleus. Now remember, the nucleus is what holds the DNA. And you see these little X things inside the nucleus? That's the DNA. We'll talk about in a second why they look like X's. But just it doesn't matter right now. The important part is that it's DNA inside the nucleus. And the nucleus, the wall of the nucleus, is actually starting to disappear. In other words, the DNA is going to be free to float around. It's normally trapped inside the nucleus, but it's going to be free to float around inside the cell and move around once the nucleus disappears. And that's important because we'll explain later, but it's important that the DNA be able to move around to different parts of the cell. So that's the first thing that happens, is that the nuclear membrane starts to disappear. And the second thing is that this stuff over here is what we call the mitotic spindle. Mitotic just refers to mitosis. And a spindle is just something that pulls on something. Okay, so this mitotic spindle is starting to form. And what this spindle is going to do is it's actually going to pull the DNA apart. It's going to rip it right in half. It's going to rip half the DNA to one side of the cell, the other half to the other side of the cell. These mitotic spindles are very important in the process of separating the cell, this one cell, into two separate cells. All right, so that's prophase. And then we go into metaphase. So at this point, you can see there's no nucleus. The nucleus has disappeared. The nucleus was here, and now it's completely disappeared. There is no nucleus. But the DNA is all lined up in the middle of the cell. The way that you remember metaphase is meta-middle. Meta-middle. In metaphase, the DNA lines up in the middle. You can see that the mitotic spindles are grabbing onto the DNA. Now, look at the DNA. I told you that it looks like an X. Well, these X's are what we call chromosomes. You've probably heard of chromosomes. Now, the chromosomes, there's more than just four. There's 1, 2, 3, 4 here. That's because they didn't draw all of them, but you actually have 46 chromosomes. Okay. And all of those will be lined up in the middle during metaphase. And your cells, the mitotic spindles, are going to grab on to each one of these pairs of chromosomes. And the point right here in the middle where they grab at is called the centromere. Now, in anaphase, after this DNA is all lined up in the middle, these mitotic spindles that formed in the very first stage in prophase and connected to the chromosomes at the centromeres, that's the part in the middle, they're going to pull the chromosomes apart. They're going to pull half the chromosomes to one side and the other half of the chromosomes to the other. So now we have half the DNA on one half of the cell and the other half of the DNA on the other half of the cell. So in anaphase, the chromosomes or the DNA, it's the same thing, are being pulled apart to opposite ends of the cell. And so now we're ready to go to telophase, the T in PMAT. And in telophase, you see this dotted line? That's the nucleus. It reappears. So it disappeared in prophase, and now it's showing up again. But this time it's showing up over half the amount of DNA that we started off with. And there's a second nucleus showing up that surrounds the other half. So now we have two nuclei, that's the plural for nucleus, inside the cell. Nucleus 1 and nucleus 2. Each one has a full set of DNA. Why not a half set of DNA? Well, remember, before we started mitosis, which is the PMAT process, we doubled the amount of DNA inside the cell. We did that during the S phase. So, really, each of these nuclei have the appropriate amount of DNA, one full set of DNA. This has a full set of DNA, and this has a full set of DNA, because we started off with double the amount. So now that we have half of the DNA, or one full set of DNA on this side, and one full set of DNA on this side, and the nucleus has showed up, has reappeared around each full set of DNA, we can now split right down the middle and separate the two halves, with each half having their own nucleus with their own full set of DNA. And that's called cytokinesis, where the cell actually gets cut right in half and separated into two cells. And then each of those two, these are called daughter cells. So each of these daughter cells, will now look like this. And they will go right back into the G1 phase, where they're just growing, developing to an adult cell. So after cytokinesis, where they're split, they each go into the G1, they grow and mature, then they double their DNA, then they prepare to go through mitosis of their own and split in half again to make their own daughter cells. Then they go through PMAT, which is mitosis, split in half, and then their daughter cells will start all over again. And that's the circle of life in your cells in your body. And real quick, I want to make sure that you guys understand the difference between mitosis and meiosis. Mitosis is what we've been talking about. It's where we start off with one cell that has 46 chromosomes, which is all of your DNA. Your DNA is not one long strand. It's actually broken up into 46 small chunks. Okay? So you don't have one big strand of DNA, you have 46 small strands of DNA inside of every single cell. And that one cell will split in half, and each new cell that we call daughter cells also has a full set of 46 chromosomes. That's mitosis. That's what we just talked about. That's different from meiosis, which we haven't talked about today, but we will talk about in the future. That's where one cell starts off with a full set of 46 chromosomes and splits in half and half again to where each daughter cell has just 23 chromosomes. Now, we'll talk about that later. I don't want to go too much into that now. I just want you to know the difference between mitosis and meiosis so that you don't get the two confused. Mitosis is what we talked about today. All right, that's it. I hope you guys learned something. And I'm going to stop now.